Welcome to another lecture of Divya Mahesh tutorial. So in the last lecture we saw the chemical structure of DNA and RNA. Then we started that various experiments for going on that the actual DNA is a genetic material of RNA. In that the Griffith experiment came. Frederick Griffith performed an experiment transforming principle in which he told that something is being transformed, that what genetic material was getting transformed. So in this experiment was performed on mice and Streptococcus pneumoniae. He took the bacteria Streptococcus pneumoniae, which causes pneumonia in mammals. So in his experiment he took Streptococcus pneumoniae, which infected mice and caused pneumonia. So in his experiment there were two strains of Streptococcus pneumoniae. One was S3 and second was R2 strain. What is this S and R? These are smooth and rough strain. On what basis? On the basis of their presence of capsule. So when he took this Streptococcus pneumonia and there was a blood agar media, so there he saw large smooth colonies were growing, which were of S3 strain type, and very few small and rough colonies were which were R2 strain. S3 strain is virulent, that it caused pneumonia, but R2 strain was non-virulent and does not cause pneumonia. It has a capsule around its surface. So like this he took, then he performed his experiment. So first, in his first round, he took S3 type and injected to mice. Mice died due to pneumonia. Next, he took R2 type strain, then injected to mice. Mice did not die. Okay, mice was alive. Next, the S3 strain was heat killed. Heat killed S3 strain. S3 strain was heat killed, denatured mice, injected mice alive, did not cause pneumonia. And the fourth, he took heat killed S3 plus R2 type strain, injected to mice. Mice died due to pneumonia. This was seen in the last one. So from the last, he concluded that something was getting transformed from the heat kill to R2 type strain, that it caused that R2 type strain to cause pneumonia. It forced R2 type to cause pneumonia. So in his experiment, the biochemical nature of what got transformed from S3 strain to R2 strain was not concluded, but he told that something is getting transformed from the S3 type strain heat kill to the R2 that caused the R2 type to be virulent. So its biochemical nature was proved by Avery, McLeod and McCarty experiment. So we saw the transforming principle, its biochemical proof for transforming principle was given by Avery, McLeod and McCarty. So they gave the biochemical nature proof of transforming principle. So in the series they took different enzymes. We'll name it RNAs, DNAs, protease. So they took these enzymes. So in every McLeod and McCarty experiment, 3 enzymes were taken. So in series, these enzymes were used. So first we'll take RNAs. When these were put with the heat kill and R2 type strain, so mice died. Why? Because RNase enzyme degraded the all RNA present in that cell. So mice was died. So it was concluded that there is not RNA that is transforming principle, that is transforming the actual genetic material. Then they took protease. So they protease, all the proteins were degraded and then was injected to mice. Then mice died. Means protein is also not that material that is getting transformed. At last they took DNAs, and when DNAs was acted, then all the DNA material was degraded and then injected mice. Then mice alive. From this they concluded that what was getting transformed, that was DNA. So the biochemical proof of the transforming principle, that the DNA is the actual genetic material that is getting transformed from S3 heat kill S3 strain to R2 strain. So Avery, McLeod and McCarty proved that DNA was that transforming principle. Then there was another experiment, Hershey and Chase experiment. So here they proved biochemical nature of transforming principle. Now time came to prove the equivocal proof, final proof for that DNA is getting transformed from. So for that another experiment came, Hershey and Chase experiment. They performed it on T4 bacteriophage virus, and which infects bacteria E. coli, Escherichia coli. So in that, this T4 bacteriophage was cultured in 2 mediums, P32 and S35. radioactive phosphorus and radioactive sulfur. Active phosphorus and radioactive sulfur. So bacteriophages were cultured. So when they were cultured, incubated, then finally when they infected the bacteria and the colonies were grown, so in that we saw that phosphorus 1 was getting transferred. Radioactive phosphorus was found in the new colony, means where they multiplied, what we say that virus which incorporated its genetic material, and the new viruses that were developed, on that the radioactive phosphorus was found, but the radioactive sulfur was not present in the next generation. So what it confirmed, that phosphorus is present in DNA and sulfur is present in protein, means DNA is getting transferred, not the proteins. So Hershey and Chase concluded that because of the transfer of radioactive phosphorus which was seen in the next generation in the DNA, but not S35, so DNA was getting transferred, not the proteins. So these were the experiments. Very many experiments were performed on what RNA, proteins, DNA, and what acted as genetic material and all those. So these were a few experiments which I discussed for you. Now thank you.